Right, so the next speaker is Dr. Mustapha Bouhrara. He's after finishing his undergraduate degree in physics in Morocco, Mustapha moved to France where he obtained his master's degree and a Ph.D. in biophysics and magnetic resonance imaging in 2012. He was a postdoctoral visiting fellow, a research fellow, and staff scientist at the NIH from 2012 to 2020. And in 2020, Mustapha obtained the Stadtman Investigator Award. He is currently serving as a Tenure Track Investigator and the Chief of the Magnetic Resonance Imaging, Magnetic Resonance Physics of Aging and Dementia Unit at the NIH. So thank you, Mustapha.
Can you see my slides, Olivier?
Yes, yes.
Perfect, thank you. Well, first of all, I would like to thank the organizing committee members of this really wonderful symposium for inviting me to give a talk here. And so the research focus of my group is centered on the characterization of cerebral changes that occur in normative aging and dementia through development and also use of advanced quantitative MRI methods. And also we are trying to understand the impact of metabolic, vascular, genetic risk factor on those cerebral microstructural changes, and also how those specific imaging biomarkers of cerebral tissue may affect cognitive impairments and also physical impairments. So actually I was planning to provide a somehow technical talk on multicomponent analysis in diffusion and also in relaxometry with a focus on Myelin water fraction determination and with some example of application, but I changed my mind at the last minute, so please bear with me here. So I realized actually that my comrades have done an excellent job over the previous talk, so I thought a more focused talk on the importance of white matter will be complementary and also to avoid redundant contents. So today I will focus my talk on myelination in normative aging and dementia, which we think that is myelin is an important aspect that is fundamental for CNS function. So probably the first thing that comes in mind when someone evocates Alzheimer's disease is plaques and tangle, and in fact histological observations starting more than a century ago by Dr. Alzheimer himself have revealed abnormal aggregation of amyloid and tau proteins in participants with Alzheimer's disease. And although the precise mechanisms of tangles and plaques formation are not fully elucidated, these two hallmarks of AD or Alzheimer's disease have long been and still today regarded to play a central role in the pathophysiology of Alzheimer's disease. But recent data are very puzzling because now we start to realize that there is a weak correlation between the amyloid load and cognitive impairment, and also more than a third of cognitively normal participants exhibit abnormal levels of amyloid and tau aggregations in the brain. But perhaps, as it has been said last day by my comrade, that the most puzzling fact is the continuous failure of the anti-amyloid drugs or interventions to reverse or at least to stop the cognitive regression after amyloid clearance, etc. So now researchers are starting to question if the amyloid and tau aggregation are really the main driver aspects of Alzheimer's disease, or are simply a byproduct of other primary drivers of this neurodegeneration. And therefore, if this is the case, perhaps the diagram shown here represented the temporal dynamic of AD biomarkers is incomplete, and probably something else that is driving these cascades of pathologies is missing from this diagram. I don't know if you can see my cursor, but let me, yeah, so I think now you can see it. So therefore, in this case, as I said, there is something perhaps missing if this is the case. And by the way, I'm highlighting this in red here, this text, because as an MRI physicist, I'm always irritated when I see that MRI is always evocated here to refer to structural imaging for volumetry in order to describe brain structure as indicated in the green curve here. So what about microstructural changes? And we all know that microstructural changes can precede macrostructural changes by decades. And in fact, if you go back and take a look on Dr. Alzheimer's papers, you will immediately notice that in addition to tangles and plaques description, he was also talking about microstructural changes, especially white matter degeneration with greater myelin loss and axonal degeneration in subjects with Alzheimer's disease in several instances of his seminal papers. But curiously, this aspect of microstructural changes have gained little to no attention until recently, where researchers start to get interested in new models, including, for example, the Myelin model of Alzheimer's disease introduced by Dr. Bartzokis and colleagues. So briefly, the reasoning behind the Myelin model is that the myelin loss might release iron, which, if not cleared, might accumulate in the brain and promote amyloid and tau formation and aggregation. I just would like to say that oligodendrocyte, the main cells in the brain that make myelin, are rich in iron because they need it as an important cofactor for myelin synthesis. So you can see here that it is plausible that the amyloid aggregation could represent a secondary aspect of the pathology behind Alzheimer's disease. And indeed, emergent studies are establishing now a link between iron accumulation and amyloid and tau aggregation, and also that iron and amyloid plaques colocalize, and that iron interacts with amyloid precursor protein and promotes amyloid beta deposition, and also binds to the tau protein and induce tau phosphorylation and aggregate phosphorylated tau protein. But nevertheless, in my personal opinion, I think that the Myelin model is still restricted and should be extended to include other aspects of neurodegeneration, especially axonal degeneration as well, which has been associated with early stages of Alzheimer's disease and also linked to amyloid and tau accumulation. So I like to call this model the white matter model of Alzheimer's disease instead of the Myelin model. So anyway, so given this, all this evidence regarding the link between microstructural changes and neurodegeneration, in my very humble opinion, I believe that this diagram that I have shown you before is likely missing the yellow part, that is the nature, the role, and the temporal etiology of these microstructural changes that could plausibly trigger the cascade of the other aspects, including amyloid beta aggregation, tau accumulation, and concomitant physical and cognitive impairment. So I think that one of our roles as MR physicists is to add this yellow line to this diagram and also define its correct position. And of course, this is a huge work that requires extensive work, obviously. So one piece of this work is to be able to map specific determinants of white matter microstructure, such as myelin and axonal densities. So as we saw throughout this wonderful meeting, there are different ways to probe myelination, but perhaps, as Dr. Lowley have explained in a wonderful manner, that probably myelin water fraction is the most specific and the direct measure of myelin content through multicomponent relaxometry. So I will not go through the physics, math, and signal processing behind this development, although this is what I love to do. But I think, as I said, Dr. Lowley and my comrades previously have done a very fantastic work to describe all different approaches that we can use to probe myelination. But what I would like to briefly indicate that in my lab, we use the method that is called the McDespot method, which combines two sets of steady-state sequences, the SPGR and BSSFP sequence. And as you certainly know, the steady-state imaging allows you to scale rapidly because you can use very short TRs or repetition times. But as you can see from those equations, this method involves the estimation of several parameters. So at the end of the day, you end up having a high-dimensional problem to solve, which makes parameter determination, especially myelin water fraction determination, very unstable. So we did some extensive work to reform the McDespot signal modeling, and including the SPGR and BSSF signal model, that we're not accounting for some duration between the excitation and the acquisition time, which is the echo time. And we using block equation, we derived like a new signal model that should be used to avoid bias in Myelin water fraction determination. And also we characterize the issues with this method, including the flatness of energy surface, but I don't have time today to go through all those technical details. And we were able to suggest a method based on the Bayesian analysis, which we call the Bayesian Monte Carlo analysis of McDespot. And as you can see here, with the BMC McDespot, you can recover maps of Myelin water fraction at clinical SNR that are very close to those derived from high SNR. And as you can see with the conventional analysis, it fails to recover the Myelin water fraction estimate in several brain regions. And just to let you know, this is a very healthy young participant. I believe it's one of my students. So the myelination should not exhibit this very low values in those regions. But I will be very happy to discuss this in more detail, the technical aspect of the BMC McDespot, and also please reach out to me if you are interested in the method or for eventual collaborations. So now that we were able to measure a myelin content, we wanted to explore the association between iron content and myelin content. And as I indicated before, the myelin model, or what I would like to call the white matter model, suggests that the breakdown of myelin might release a great store of iron that could promote amyloid and tau aggregation, etc. But the main question is there a link between iron content and myelin content? So to investigate this question, we implemented our method for myelin content, which is the BMC McDespot for myelin water fraction determination, and also the susceptibility weighted imaging for mean phase determination, which is a semi-quantitative metric of iron content with lower mean phase values have been associated with higher iron content. And of course, as we saw here, there is much more sophisticated way, including the QSM method that we are very interested in, we are looking at. But at that time, this is what we had on our hand. So we correlate our iron measures with myelin content or myelin water fraction, accounting for covariate. And as we can see here from this plots, iron content or deposition increases with decreasing myelin content. And this association was significant in most brain regions investigated. So with this, and this is cognitively normal participants. So with this evidence, and I believe there is more evidence as we saw in multiple sclerosis and other neurodegenerative diseases of the association between decreased myelin content and increased iron content, it is very plausible that the amyloid and tau aggregation can simply be a byproduct of other underlying mechanisms, including, for example, iron accumulation due to myelin breakdown. I'm not saying this is what's going on, but this is something that needs to be investigated, including in Alzheimer's disease, and also using histological measures, which may lead at the end of the day to markers of disease progression and treatments, including, for example, iron chelation or myelin enforcement. So now the natural question that comes to mind is what causes myelin breakdown? So we looked to several factors, and obviously the first and the main risk factor is age. And our results showed that myelin increases from young to middle age, followed by a rapid decrease afterwards in most cerebrum and brainstem structures. And also our results indicated that the interior regions, including the frontal temporal lobes, are the first to demyelinate as compared to the posterior region. And this seems to be in line with the retrogenesis paradigm, which indicates that early myelinated regions, which are the posterior regions, such as the occipital lobes, are spared from neurodegeneration as compared to the interior regions. And interestingly, what we saw also is that the brainstem exhibits earlier peaks of myelination, and this is kind of expected because this structure represents a structure for survivals and vital functions of the body. So like breathings and all those and cardiovascular function and all that stuff as compared to the cerebrum. But unfortunately, we believe that this is an overlooked structure despite the convincing evidence of its early involvement in several neurodegenerative diseases, including Alzheimer's disease, but we are keeping a close eyes on this structure. So we also looked at genetic risk factor, and especially the IPOE gene, and I'm sure that you are aware about the fact that the IPOE isoform 4 is the highest genetic risk factor for Alzheimer's disease, right? And there is a debate also about the isoform 2 that have some protective effect against neurodegeneration. And also we know that the IPOE facilitate the transportation of the cholesterol and lipid, which are the main constituents of myelin. So we thought that there is probably some relationship between the genetic risk factor and myelination. So we applied this analysis in, again, those was cognitively normal participants without any major neurological problem or health problem. And what we saw is that the carriers of the IPOE isoform 4 have much lower myelin as compared to non-carriers. And also the IPOE or carriers of IPOE isoform 2 have statistically significant higher myelin content as compared to non-carrier. And this shows also some evidence of the relationship between myelination and the highest risk factor of Alzheimer's disease. So we also looked at metabolic risk factor, and specifically obesity as measured by the waist circumference and also body mass index. And what we saw is that participants affected by obesity exhibit much lower myelin content and significantly much lower myelin content than lean participants. And fortunately, this is a risk factor that we can reverse through diet and exercise. And we are very interested to look at whether inverting or fixing this metabolic dysfunction can reverse or can lead to remyelination or normal levels of myelin in this participant. And this is what we're planning to do in my lab. But the main focus, one of the main focus that we are aggressively looking at in my lab is directed toward the vascular risk factor, and especially cerebral blood flow as the end product of all those vascular risk factors. So we believe, and actually it has been shown by Dr. Bogdan's Dragansky, I believe 2 days ago, that cardiovascular risk factors are the main aspects affecting brain health. And indeed, in the recent years, there is mounting evidence demonstrating deteriorations of cerebral tissue with aging, and to a higher extent with neurodegeneration, of course, as well as deficits in cerebral blood flow. So one of our main hypotheses is that since myelin maintenance through oligodendrocyte metabolism is an energy-demanding process, this metabolism becomes particularly sensitive to hypoxia and hypoperfusion. In other words, with lack of nutrient, oxygen, glucose to the oligodendrocytes, it becomes difficult to support maintenance of myelin. And also adequate blood flow is crucial for removal of metabolic byproducts, including iron accumulation and amyloid and the other protein aggregation. So I like to see the brain as a house. You need to have a clean water coming in for normal functioning, and also to get rid of the bad water. And if any dysfunction can happen in one of these two processes, you can imagine the disastrous consequences that can happen. So here I'm showing some of our results of the association between myelin water fraction and cerebral blood flow. I just would like to mention we measured CBF using the ASL, specifically the PCASL, the pseudo-continuous arterial speed labeling. And also I'm just putting here some references of some post-analysis methods that we use to improve determination of CBF values if you are interested in. So as you can see from the regression plot, a lower myelin content is associated with lower cerebral blood flow. And as you can see from the table, this association was significant in most brain regions investigation after accounting for several covariates, including age and sex. And this novel result provide, I believe, the first demonstration of potential association between cerebral blood flow deficits and myelin decline in cognitive normal subjects, and also highlights the importance of blood supply to the brain for myelin homeostasis. But obviously, there is drawbacks to our study. And the first one is the limited cohort size and also the cross-sectional nature of the design of our experiments. So for that, we expanded our analysis to analyze CBF, or cerebral blood flow, and DTI data acquired in the CARDIA study, which is a large longitudinal cohort of well-characterized adults that examines the development and determinants of clinical and subclinical cardiovascular disease and the risk factor. So using the linear mixed effect model shown here, we specifically looked whether the cerebral blood flow at baseline could predict changes in DTI indices over 5 years, and those indices are fractional anisotropy and mean diffusivity that were available for us after correcting for the covariate displayed in this slide. So the result clearly shows that participants with low cerebral blood flow at baseline, which are indicated with the red curves here, exhibit more rapid increase in mean diffusivity and steeper decline in fractional anisotropy. In other words, more rapid decline in microstructural integrity, as compared to participants with higher cerebral blood flow at baseline, shown in green. And interestingly, this association were mostly observed or were statistically significant mostly in white matter regions, which clearly supports and agree with our previous study, and also, as I said before, with Dr. Dragansky's result that cardiovascular risk factors seem to affect more myelination than other aspects of cerebral tissue, and this is very interesting. So all these findings suggest that the blood supply to the brain may be an important determinant of cerebral tissue health, and also a potential therapeutic target of neurodegeneration. But of course, more work is still to be done, especially in the context of MCI, mild cognitive impairment, which could be a prodromal phase of Alzheimer's disease, and also in dementia, and this is underway in my group. So now, who cares, right? And who cares if white matter or myelin degenerate as long as we can stay cognitively sharp and physically operational, right? So let's see whether white matter degeneration, especially white myelin degeneration, could affect cognition, in which case we definitely should care. So in a beautiful demonstration conducted by Zhao, my junior collaborator, who will be presenting shortly after me, another outstanding work of him on one step closer to real-time myelin water fraction using deep learning analysis. And this is in collaboration with Dr. Murad Bilgil, one of our brilliant minds in the NIA. What he showed is that in a cohort of over 120 cognitively normal subjects, and using the linear mixed effect model shown here, that lower myelin content was associated with steeper decline in cognition, including executive function, verbal fluency, memory, and attention. In red, those are participants that have lower myelin content, and in green, those participants that have higher myelin content. So, and for those participants, we had the retrospective longitudinal psychological testing. So we look at the retrospectively on the changes when we anchor the myelin water fraction measures as a baseline. And this finding highlight the importance of white matter and especially myelin integrity and cognitive functioning, and suggests myelin water fraction as a potential sensitive and specific imaging biomarker related to predicting cognitive changes in normative aging, and perhaps in cognitive impairment. This is something that we are looking to as well. And also, as I said, motivates the study to examine this relationship between myelin deficit and cognitive impairment in MCI and also Alzheimer's disease. And I believe this studies might lead to more rational designs of therapeutic interventions, including, as we said before, myelin enforcement. So what about physical functioning, right? And here briefly, we looked at the implication of myelination on gait speed, which is an integrative measure of physical health or functioning. Again, this is a cohort of cognitively normal participants, and this work is under preparation by my brilliant student, Mary Faulkner. What she clearly shows here is that participants with low myelin content exhibit lower rapid gait speed or usual speed gait. Usual speed gait, rapid speed gait is the same thing. We ask the participant to walk in a corridor for usual speed gait, gait speed at their usual speed. And for rapid gait speed, we ask them to walk as fast as possible without running. And again, this finding highlights the importance of white matter, specifically myelin integrity in functional, in physical functioning. If I have a few minutes, I just would like to touch on work that has been conducted with my collaborator, Dr. Keenan Walker at the NIA, where we look at the myelin and axonal density. Axonal density, we use the NODI analysis to measure the NDI, the neurite density index. And we look at the association between myelin water fraction and NDI versus the Alzheimer's disease and neural injury and astrogliosis plasma biomarkers. And although the relationship between myelin content, axonal density, and Alzheimer's disease now is more and more established, less is known about the extent to which this white matter features relate to distinct component of Alzheimer's pathogenesis, particularly in the prodromal phase or the preclinical phase of the disease. So by examining this relationship between myelin content, axonal density, and biomarker of Alzheimer's disease and non-specific features of Alzheimer's disease, including, for example, neural injury and astrogliosis, we can better, I believe, understand how this distinct facets of the Alzheimer's process relate to white matter degeneration. So rapidly in this study, cognitively normal participants again, we examined the association between plasma biomarker of Alzheimer's pathology, the amyloid ratio, amyloid beta ratio 42/40, and p-tau 181, neural injury measured through NFL, and reactive astrogliosis measured through the GFAP. And we correlate this to our measure of myelin water fraction and NDI, as I said, derived from the NODI analysis. And our results clearly indicate that higher GFP were associated with lower myelin water fraction. And also this association was close to significance for amyloid beta ratio in our secondary analysis that I'm not showing here. And also what we saw is that higher levels of NFL and GFP were associated with lower axonal density, of course, after covariate analysis, covariate adjustment that I'm displaying here. And also we found in a secondary analysis in some specific regions in the brain, there is some association between lower amyloid ratio 42/40 and higher p-tau that were associated also with lower axonal density. And clearly this study shows that axonal loss and demyelination might occur with neurodegeneration and astrogliosis ahead of clinical meaningful cognitive decline. And also, we believe, stay the foundation for further investigation, including longitudinal assessments, which we are trying to do in our group, to clarify the extent to which plasma biomarker of AD and neurodegeneration pathology reflect biologically meaningful changes in white matter structure and function defined using the specific MRI measures. And again, probably those microstructural changes might precede, as I said in the beginning of my talk, this amyloid and tau aggregation that we are seeing in Alzheimer's disease. I wanted to talk about our work on axonal density, but for sake of time, I'm just going to jump to the conclusion. So I believe we addressed some central questions here in the relationship between brain microstructure, aging, and dementia. We believe that demyelination, axonal damage, I didn't have time to discuss it, but also iron deposition and local blood flow may be key factor aspects in neurodegeneration and early biomarkers of neurodegeneration. And of course, as has been discussed heavily here, is this will provide motivation for development and also the improvement of the methods that we are developing. Of course, our method, all of them, all quantitative MRI methods are based on simplification and model assumptions, and also does not account for different physiological and experimental factors. But this is what keep things fun and keep things interesting to improve those methods to make them more specific and sensitive to the microstructural changes and become much more specific and sensitive to capture those changes with aging and also with pathology. Of course, this work will not see the light without the contribution from my previous students and also my current students and collaborators. Thank you very much.
Thank you very much, Dr. Bouhrara. A fascinating talk. Aurélie, is there any questions in the chat?
Yes, so I'm going to actually start with a question that was previously asked for Cornelia, but I think it fits also your talk perfectly. So the question was from Zayon Gong. who asked, what do you think it takes to push this wonderful technique to the clinical trial stage as a diagnostic tool? I am not aware of any clinical trial at this moment.
This is a fantastic work. This is something that we are always discussing, and I think there are different aspects to make those techniques. First of all, there is a huge validation that has to be done through histological validations, and also there is a standardization aspect that we have to do through multi-vendors. And I believe in the diffusion work, this work has been done or start to be done, but in the relaxometry and specifically Myelin water fraction, we are kind of behind a little bit. But this is, I think, two aspects that fundamental before like putting those techniques to the clinics is really the validation and also the standardization of those methods, because again, those methods depend on the experimental parameters. So if you want to compare studies, etc., they have to be designed within the specific or the exact experimental parameter. And also there is another aspect, which is the open source codes and collaborations, and those methods have to be applied for different studies to prove these values. And again, I feel like in the diffusion work, they are doing a fantastic work. And this is something that as relaxometry people here, we need to learn from the diffusion guys and try to mimic that. But this is an excellent question.
Thank you. So we had also another question from Christine Tardif. So she was saying, thank you for the great talk. You showed several applications looking at white matter pathology. Are the acquisition techniques advanced enough to look at intracortical myelin?
That's a very good question. And that's we can look at that definitely, but I don't think the cortical regions exhibit very low myelin, for example, content, and that makes when you have a short fractions in your models, makes the determination or the estimation of the short fraction when they are very low, very, very sensitive to SNR. So idea will be how we can improve the SNR, those images. In theory, it could be done, but we still need to improve our methods to be able to capture those very, very low, amount of myelin that is in the cortical regions. And also there is other aspect that has to be addressed. And we are seeing some work, very fascinating work done in this aspect, which is the contamination from the CSF in those regions that has to be carefully taken care of. So in theory, yes, but in practice, we still have a road there to do. But it's something really that we have to look at for sure.
Thank you. So we have another question from Dr. Sridhar Narayanan. So loss of diamagnetic myelin will lead to increased phase shift on susceptibility weighted imaging. How did you decouple this effect from that of iron deposition?
That's a fantastic question, and it's true. But what happened here when we saw is it should fight against the trend. So despite this increase in the phase shift, we still see negative values with the iron, and the myelin should exhibit the opposite trend. And also another thing that we observed that I didn't show here is that when we look at the mean phase value versus age, we saw a very linear relationship with age, but with myelination, we see this inverted U shape. So there is some aspect that they are indicating that they are decorrelated in some way, but they are not like probing the same aspect, but certainly there is some contamination there that has to be addressed, and we have to be aware of it as discussed in our paper. But this is an excellent question.
Okay, thank you. I'm going to take just one last question from Julie Autois, who says, great talk. Is reduced myelin water fraction in the AD continuum linked to higher level of white matter hyperintensities on T2 flare MRI?
This is an excellent one. I think that has been shown. I just reviewed the paper, or not reviewed, it's from our lab that they are showing this, but not using myelin water fraction. I think using other techniques. I'm not aware, but again, I'm not a specialist of Alzheimer's disease, but probably there is some work there that we have to look at, but this is an excellent question. This is something that we really would like to look at in our in our group. The only problem is the availability of large cohorts of Alzheimer's disease to really explore this question in a convincing details. We didn't look at it yet, but that's something that has to be investigated for sure.
Thank you so much. I think we're going to move on with the program. We are a bit behind.